Well, actually, I've retired from the University of Georgia as of December 31st. Started getting children of my first students coming through, and probably not too far from the grandchildren. It's just time for me to check out. So I'm going to talk today about seed quality and endophyte testing. You know, I think there are some pretty good cases presented as, you know, if you have a field that you're getting ready to renovate, this would be an example. What are you going to do? And hopefully after today, we've convinced you that you ought to be thinking about novel endophyte tall fescue and not endophyte-free tall fescue. So this is a picture. These are side-by-side plots at the University of Georgia. This is Kentucky 31 endophyte infected, the old standby. This is Kentucky 31 endophyte free. And this is back before we had some really rapid systems for testing, but I'll almost guarantee you that that plant right there, which is as green as that plant over there, is infected with endophyte. That was probably a low endophyte population of tall fescue and not necessarily endophyte free. But you can see here that, you know, the endophyte free will go out of pasture in preference to the endophyte infected. Okay, so you need to know when you go down to your seed dealer that when you purchase novel endophyte seed that you're getting what you want. And if you look at what's on a bag of seed and what is required by law to be posted on a bag of seed, it's this type of information. The species of the seed, what you are going to be planting, a lot number that's on there, what the germination percentage might be, the purity of that seed, any other crop species that might be in there, like annual ryegrass, noxious weeds that are just weeds that are difficult to control, so you don't want any of those, because if you're planting noxious weeds, you're just planting potential problems, okay? So we don't want any of those guys in there. Any inert matter, which would be the chaff, things like that. Other weed seed that might be in there. The origin of where this seed came from. Any seed treatments that might be on there, fungicide, insecticide, and so on. And then a test date as well. Oh, okay, stop. Thought I was going to do a dance here. All right. So a normal seed test is information that's provided on a bag of seed that's required by law, okay? Claims of endophyte are not required by law except for, I think there's one exception to that now, and that is in the state of Mississippi, if you are going to sell endophyte-free seed, it has to be labeled as endophyte-free seed. And when they say endophyte-free seed, they mean less than 5% of toxic types in the seed. Okay, so there's no regulation whatsoever for labeling for novel endophyte technology. And that's been a problem. So let's just review what the endophyte is. It's this little curlicue organism that's living in between cells. It doesn't invade the cells. It's not a pathogen like a lot of other fungi are. It's hanging out in between these cells, just scavenging nutrients off of the cells. And the plant is giving this endophyte a happy home. And in return, the endophyte does some things for the plants, like drought resistance, insect resistance, in some cases, disease tolerance. If it's toxic, it provides resistance to mammalian grazers that are out there. We talked about the toxins being high, low in the profile of the plant, and the animals graze down and they get more toxins, and that's the signal to stop grazing, okay? So it does a lot of great things for the plant. And if we remove the toxicity, you get all those benefits that are associated with the other stresses that we talked about, but you've removed the toxins to the plant, which means the animals are going to eat more, and we've talked about that. This is just another picture here, similar to what Carolyn showed you. Okay, so the life cycle of the endophyte, well, it starts out in the seed, and as this little leaf develops into what we call a tiller, And we put tillers on your tables there. So this is a tiller right here, or a stem. We don't really call them stems in grasses, but it's a stem tissue-like thing. It's going to invade that. It's going to take about 6 weeks for that endophyte to show up in these seedling plants. Okay, so it's down here, and it's actually going to invade these seed heads. Right now, down below the soil surface around here, the plant has shifted from vegetative growth, so it's not going to produce more leaves on this particular tiller, but way down below the soil surface, the plant has shifted from vegetative growth to reproductive growth, and there's just a microscopic little seed head down there, and the endophyte is invading that right now. And then as the seed head grows up, okay, the endophyte is already deposited into the seed, and then you harvest the seed and it just regenerates itself through that cycle. So endophyte infected seeds are going to produce endophyte infected seeds. Toxic endophyte infected seeds are going to produce toxic endophyte infected seeds. So the seed is the vector of dissemination that we've already talked about. An important thing for you to understand is that the genetics of the endophyte in there, if it's a non-toxic endophyte, it doesn't convert back to a toxic endophyte. Okay, so non-toxic endophyte infected seed will always produce non-toxic infected plants that are going to produce non-toxic infected seed. Okay, so we already covered that. So when I first started out as a faculty member, we surveyed a bunch of pastures, and we found that 95% of all Kentucky 31 tall fescue pastures were infected at high rates. We're talking 60, 80, 90, 100% infection. Okay, every once in a while you'll find one that's not. These endophytes, they talk about ergot alkaloids. Ergot alkaloids are cousins or relatives of the hallucinogen LSD. So your animals are consuming these toxins that are very similar to LSD. And if I had more time, I'd tell you a good long story about that. But anyhow, not me. So these novel endophytes do not produce the toxins. And we can remove the endophyte, Craig's favorite slide. Okay, take the toxic endophyte, make endophyte-free plants, and then with some laboratory trickery like Carolyn can do, put these non-toxic endophytes back into the tall fescue plant and then generate the products that these companies have. Another thing that along the development of all these non-toxic endophytes, we found that not all endophyte and plant combinations are necessarily good. So early on, some of the work that Joe Boughton did found that you could take some of these endophytes and even though they're non-toxic, insert them into a tall fescue plant, and you might only have 30% transmission into the next generation. Okay, and all the products that you're going to hear about today, they've gone through the process of selection and making sure that the endophytes that are in their tall fescue varieties do pass to the next generation. Okay, so it's kind of a selection process. It's not just automatic if you just have a non-toxic endophyte that it's going to make a good marriage with the forage variety. There's some other things that can control the amount of viable endophyte in seed, and that is harvest conditions. If they harvest when the seed is too green, the viability of the endophyte is compromised, and then not only that, but you take that green seed and put it in storage, and the endophyte will die in a preferential rate compared to one that's been harvested at physiological maturity. And then storage conditions, temperature and humidity are the big enemies that we have to work with. So if you have high humidity or high seed moisture content, the endophyte dies at an accelerated rate. And if the temperature is too high, the endophyte will die also. One of the things that we always warn people about is don't go buy a bag of seed, put it in the cab of your truck, go down to the coffee shop and hang out for a while. Take that seed home, get it in good storage conditions so that you're not losing viability on your endophyte. Okay, so how do we go about testing to make sure that when you go down and buy a bag of seed that it's actually what you expect it to be? And this is just an example of a system that we have developed. It's based upon antibody technology or antibody chemistry. And you probably don't understand what that is, but I can give you a practical application that many of you have had in your life, a home pregnancy test. Okay, that is antibody chemistry. So we use the same technology, a little bit different format than you would have in a home pregnancy test, but same technology. And I've got a little demonstration up here is how we go about doing this. We just put seed on a membrane and we extract the things that the endophyte or that the antibody detects on that little membrane right there. It's not all that sophisticated demonstration, but it's to show you how simple it is. Okay, and then we go through a series of solutions that have got some different reagents in them. And when we have endophyte presence, we see something that looks like this. So here's a seed right here. Here's a seed right here, and it basically the color reaction mimics what the shape of the seed is. So we can detect this right here, for example, is a seed lot that tested 91% infected. Here's a seed lot over here that tested 0% infected. This particular test is an international standard. We sent this test kit out around the world, and we had people that were trained to look through the microscope and detect the endophyte through the microscope also run it with this test. Turns out that this test is more reliable than the microscope is, so it's now been accepted in 2001 as a standard international method. So we feel very comfortable, very confident about the technology we've developed. So this is in seed, okay? Here's what it looks like when we're testing stem tissue. So remember I told you that the endophyte is going to grow into a developing tiller, okay? That's how we test for viability. Just because the endophyte is in the seed does not necessarily mean that it's viable. So we come back after we've grown a tall fescue plant for 6 weeks, and we harvest the base of the tiller. We make a little wafer, cross-section of the stem base or the tiller base, and we lay it on our membrane. We extract the proteins out of it the antibodies are sensitive to, and we use a little different color reaction, but this is what you see. These pink circles here are stem bases that are just sitting on that membrane. And here are stem bases in which there's no endophyte in that tall fescue plant. Okay, so we know we can test for endophyte in the seed. We can test for viability in seedling plants that we grow. How about the toxicity? That's another big component. That's probably the biggest component, most important component. And again, we use antibody chemistry, a little bit different format than what we showed you before. The antibodies that we developed that we're testing for toxicity with, we actually took those antibodies and injected them into cattle that were suffering from fescue toxicosis, and we reversed the toxicosis condition within 30 minutes. So we know that we've got an antibody that tests positive for the toxins, okay? So the way it works is that the color reaction that we get, well, let me show you also, this is just on a little plastic plate like this right here, okay? And each one of these circles that you see right here is like a little bowl or what we call a well in this plastic plate. And we can take seed and put seed into each one of these wells, and we can extract the toxins out and use our antibody test to detect the toxins. So when we get a yellow color develop like that, that's an indication that the seed is non-toxic, okay? If we get a clear well developing like that right there, that's an indication that there's toxins present in the seed. So Ray talked about 400 parts per billion as kind of being the level at which we start seeing toxins, or what we call fescue toxicosis. We have seen values almost 100 times that high in pasture samples that have been tested, not here in Kentucky, but another state. To give you an idea of the sensitivity of this test, so 400 parts per billion is when you start seeing toxicity. That clear color occurs at 25 parts per billion. So if it's testing yellow, we know that it's below 25 parts per billion, and we consider that to be non-toxic then. So it's very easy for us to just extract these toxins from individual seeds and get a percentage of off types that you might have. So in addition to seeing all this information that we've talked about that are associated with what's legally required for these seed companies to put information on the bag, they have adopted this logo right here that has to go through the testing protocol that I just described to you to document that it is non-toxic. And the requirement is that the endophyte be, the viability of the endophyte in the seed be 70% or more, and that the toxic types have to be less than 5%. I've been testing since 2001 now, so over about 18 years, and we have not seen toxic types coming through on these novel endophyte products. In other words, the seed companies are really doing a great job out in Oregon where they produce this seed of making sure that they clean up their fields, that they don't have any mishaps when they go harvesting, so that when the seed comes to me, it's clean, the label goes onto it, you look for that label, and then you've got endophyte, you've got what you want out in your field. So looking for the label, the logo is the important part for you to do that's telling you that it's gone through this testing process and met the requirements that are necessary. And with that, I'll answer any questions. I know it's lunchtime.
I have a question for Nick.
Well, it's either that logical or that boring. Yes.
Are those kits that you're talking about very expensive?
So this kit right here is, I think it's $250 now. We actually don't sell the test kit for the seed because we want everybody, the Alliance wants everybody sending the samples to our lab. That's a consistent thing for them to do. The endophyte test in the seed and the endophyte test in the plant tillers, I think the seed kit now is about $80 and the plant tiller kit's $90 or $95, something like that.
Is there any other questions? Briefly, good story. What's the cost of the five?
That's going to vary from seed lot to seed lot. I've done some storage testing like that, specific for companies, and I don't want, I'm kind of obligated not to say what we found because it's their product that we tested, and so I'm reluctant to say that. If the seed companies want to say anything about that.
We've done a lot of storage trials, we were looking for the right kind of bag.
Yeah, we did test some seed, and I won't say which company it was recently, and it had been harvested last year, and it tested out at about 85% infected. There were some other seed lots that tested lower, but there's some that tested 85%. So that's why I say it varies from seed lot to seed lot. But I can tell you that they will not sell anything knowingly that does not meet those requirements up there.
Most companies, bulk seed, and it stays wet, you can't get it planted. Most of them will trade out fresh seed for you. So definitely Cobra seed or something you can store for years on your own. Still be pretty good. This novel endophyte seed, you want to plant it the fall after it was harvested or the fall of the spring, even with ideal storage conditions.
So if you're interested in testing your pastures, Craig mentioned test to validate that you've got the toxic pastures. If you've got old pastures, 40 years old, 30 years old, something like that, I can guarantee you that if they're infected with the bad endophyte. But I've got some pamphlets up here on endophyte testing. It's got our company information. It doesn't have our address. It has our telephone number and it has our website. If you want to submit a sample, just, contact us. We'll give you the street address, but, we're talking about LSD type compounds, and we have to have a DEA number to have some of those compounds around in the lab, and the lawyer told us, man, don't put your address out there because you'll have people breaking in.
All right, let's thank Nick for his presentation.